we are going to talk about influenza virus. Influenza virus is a orthomyxoviridae virus. It's a myxovirus, and influenza virus is the orthomyxoviridae का जो फैमिली है उसके अंदर इन्फ्लुएंजा वायरस पड़ता है. पैरामिक्सोविरिडे के अंदर अलग अलग पढ़ता है, लाइक मम्स पढ़ता है, पैरा इन्फ्लुएंजा पढ़ता है, वट एवर. बट आवर सिलेबस इज मोस्टली कंप्राइज्ड ऑफ ऑर्थोमिक्सोविरिडेज इन्फ्लुएंजा वायरस. इट इज़ वेरी इंपॉर्टेंट. सो सम ऑफ द बेसिक थिंग्स दैट वी हैव टू नो इज what is influenza virus. So influenza virus has influenza A, B and C, which is very common, and a D variation also. There are 4 genera of this virus, and morphology wise you don't have to know anything. The least you can know is that these are RNA viruses. Influenza is an RNA virus. Most of the viruses are RNA virus, but there are certain DNA viruses too. Certain DNA viruses are like herpes and herpes, adenovirus. There's something happy, some shit like that. Okay. Anyways, papilloma virus, I think, whatever. I don't know. Human papilloma virus is different. I'm sorry. That is different. But anyways, so what we have to know about influenza A and B is that influenza A and B consists of 8 segments of RNA, and influenza C and D consists of 7 segments of RNA. These are not that important. Main important thing about this virus is its morphology. के ऊपर, like viruses के body के ऊपर, there are 2 things. The envelope, okay. The envelope, it consists of a lipid envelope in which 2 types of glycoproteins are inserted. This is very important. This thing you have to know. 2 types of glycoproteins are HA and NA. HA is heme agglutinin and NA is neuraminidase. So basically heme agglutinin जो होता है, it helps in the entry of virus into the host, okay, or into the cell or whatever. So it is more in number, and it has it can attach to the sialyl acid receptors or residue, whatever. Neuraminidase जो होता है, it is less in number, it is mushroom shaped, and it actually helps in the degradation of the sialic acid receptors. It does not facilitate viral entry, rather it helps it in exiting. Okay, it degrades the sialic acid receptors, and that one helps in the attachment to the sialic acid receptors. It binds to the sialic acid receptors. HA binds to the sialic acid receptor, NA does not bind and rather degrades it, so that detachment होकर exit हो जाता है. So this is one thing, and according upon based on the ribonucleic proteins and M proteins, influenza viruses are of 4 types: A, B, C, D. Okay. Now subtypes, okay, the influenza subtypes, basically, see, I'll tell you something. Influenza का जो types होता है ना, it is all based on HA and NA, that is heme agglutinin and neuraminidase. Okay, neuraminidase. So subtypes तो बोला मैं, 4 type का होता है, 4 genres होता है. So influenza A has यहाँ पे there's a lot of information given, like 18H subtypes and stuff like that. You don't have to know all this. You have to know influenza A is there. Influenza B viruses are not usually classified into subtypes. Influenza C जो होता है, it is responsible for mild infections. Okay, and D is not pathogenic to humans. It only causes influenza in cattle. This is all you need to know about from this phase. Then the most important part is the antigenic variation of influenza. It is the most unique property of influenza viruses, which is due to the result of antigenic changes occurring in hemagglutinin and Na. Okay, papillomas. Basically, what is happening is antigenic variations जो होता है, there are two types. Okay, and यहाँ पे बहुत important सा question बनता है जो हर एक पेपर में देगा, that is antigenic drift and shift. Now, what is an antigenic drift? It is a minor change occurring due to point mutation in the HA or NA gene. Point mutation, resulting in alteration of amino acid sequence of the antigenic sites, resulting in the alteration in the amino acid sequence of the alteration sites on HA or NA, such that the virus can partially escape recognition by the host's immune system. Like your host can't recognize it. That's it. So basically it is mostly seen in influenza type A and B both. Okay, it results in outbreaks and minor periodic epidemics. Okay, it's a gradual change, and this antigenic drift thing occurs every 2 to 3 years. Now, antigenic shift is quite different. It is an abrupt or major drastic. It's a sudden thing that happens. It's an abrupt, major, drastic, discontinuous variation in the sequence of the viral surface proteins, HA or NA. Again, sab HA or NA ka khel hai. That occurs due to genetic reassortment between two different strains, between genomes of two or more influenza viruses infecting the same host cells. ठीक है, रिजल्टिंग इन अ न्यू वायरस स्ट्रेन. मतलब दो डिफरेंट इन्फ्लुएंजा वायरस का स्ट्रेन है और जीनोम्स है, वो ये सेम होस्ट सेल को इन्फेक्ट कर रहा है. ठीक है, दो आरएनए सेम होस्ट सेल में इन्फेक्ट हो रहा है somehow, and which results in a new virus strain altogether. This which is completely unrelated to the predecessor strain. मतलब it's completely unrelated to the previous ones. Okay, thus antibodies developed against previous strain are very ineffective, so it is a totally new thing altogether. It occurs only in the influenza A virus और इनके subtypes में होता है. There are actually influenza A type का बहुत सारा subtypes होता है. There are 18H subtypes of influenza A and 11N subtypes. This thing you might need to know. So H1 to 18 होता है, N1 to N11 होता है. That's it. You have to know this, not gonna lie. And it occurs in influenza A virus and results in pandemics. Okay, like causes major epidemic and pandemic. And one of the example is the H1N1 pandemic of 2009, and it occurs in 10 to 20 years, once in 10 to 20 years. This is one of the most important questions. इसमें तो तुमको 4 marks का या 3 marks का दे ही सकता है. Give reason why. Anyways, pathogenesis देखा जाए तो influenza दे सकता है. See, it won't be a 10 marks question honestly. Influenza can never be a 10 marks question. It will be a short note obviously. Lab diagnosis में short note दे दिया या इस antigenic shift or drift में short note दे दिया. Pathogenesis हो गया. तो pathogenesis में see, first of all, influenza transmission is by inhalation of respiratory droplets. Simple. Okay, coughing and sneezing or via like contact with surface, okay, which is infected like that. Target cell entry. Okay, now the viral HA hemagglutinin attaches to the sialic acid receptors on the respiratory mucosa, and then that leads to the viral entry that I already told you. Multiply locally होता है. ठीक है, infected cells में locally multiply होता है within several hours. Actually इसका जो incubation period है वो भी कम ही होता है. It takes around 18 to 72 hours. Okay, and then it spreads to the lower respiratory tract, or sometimes it might spill over to bloodstream also, which can result in extra pulmonary damage. ठीक है, so yeah, and These are the things. Local damage में basically तुम्हारा desquamation of superficial mucosa हो जाता है of the respiratory tract. Desquamation as in squamous cells जो होता है वो desquamation हो जाता है. That is, the squamous epithelium is getting destroyed, and cell destruction होता है in the upper respiratory mucosa, okay, superficial. So yeah, that's it. You don't have to really know how the body reacts to it, but anyways, so yeah, clinical manifestations mein you have to know about the this thing, like the incubation period I told you. Okay, the reservoirs of this are actually animals, birds and humans only. Humans se bhi human spread hota hai, and everything I told you. Uncomplicated influenza or flu syndrome is one of the manifestations that might occur. Majority of the individuals are either asymptomatic when they get influenza or develop minor upper respiratory systems such as chills, headache, sore throat, cough, myalgia, high grade fever, anorexia. It is a self-limiting condition and often undistinguishable from the infections causing by other respiratory tract infections. So you don't know whether you have influenza or not, but certain times serious complications can occur. This influenza thing is very easy, okay, body can easily detect it and it's not a huge deal, but sometimes other complications like pneumonia can happen. Now once pneumonia happens, you have secondary, like most common complication is pneumonia, where influenza virus patients second, like basically what happens is, as I said, no, कि respiratory mucosa damage हो जाता है. So what happens is, if you are suffering from, if you got influenza virus attack, then agents such as Staphylococcus aureus, right, Staphylococcus and pneumococci, all these things can actually penetrate. Okay, they actually are present, as I said in previous videos also, they are present as normal flora in your upper respiratory tract, in your nasopharynx. So they can just get inside. Haemophilus influenza can also get inside, and they can cause a influenza pneumonia type of a syndrome. Okay, it's rare, but it can be caused. And sometimes chronic bronchitis, COPD, okay, just all the lung related shit, all the सांस फूलने वाला चीज़ सब लिख दो यहाँ पे, ठीक है. एक्स्ट्रा पल्मोनरी कॉम्प्लिकेशन में यू कैन राइट अबाउट मायोकार्डाइटिस, एनसेफेलाइटिस ऑलमोस्ट एवरीवेयर. ओके, रेस सिंड्रोम बोल के एक चीज़ होता है, दैट इज़ वेरी इंपॉर्टेंट. दैट इज़ जमशेदपुर फीवर काइंड ऑफ अ थिंग. It is actually a fatty degeneration of liver with acute encephalopathy occurring in children and adolescents like 2 to 16 years of age. So this is something that happens. It's not really significant, but okay. Now what I have to tell you about is the lab diagnosis. Okay, there's also something called an avian flu, which is a birds are the primary reservoir of this influenza virus. It's a kind of avian thing only. It spreads from birds, this influenza. We will come for the lab diagnosis of influenza. Okay, wait, what? Yeah, we'll come for the lab diagnosis of influenza virus rather. So since there's a virus, we have to do a specimen collection of it. So nasopharyngeal swab will take. Okay, it can be a polyester tip, so you can take a nasopharyngeal swab. Then it has to be it has to be refrigerated at 4 degrees Celsius apparently. Okay, and this thing you have to mention. Then lab diagnosis mein you have to there's something that you need to say is that isolation of the virus has to be done. Okay, the isolation, and also there's a transport medium for this. Once you take it, right, you store it in VTM. Okay, that is a viral transport medium, and then you do an isolation of the virus by embryonated eggs, that is amniotic cavity, embryonated eggs, and primary monkey kidney cell lines. Primary monkey kidney cell lines is the most important one, have been the best method of choice for isolation of influenza virus in the past. Basically, it is done by heme adsorption or heme agglutination test. Okay, basically the test is heme agglutination test. Now after that we go for molecular method is okay, that is the best one honestly, and RT-PCR is done. Okay, and real-time RT-PCR is also reverse transcriptase polymerase chain reaction. It's highly sensitive and specific, and real-time is also done. Biofilm film array thing is also done. Respiratory panel, that is not that important honestly. Direct immunofluorescence is also not that important for this. The treatment of this would be, treatment of influenza virus would be neuraminidase inhibitors. Okay, these are actually a viral thing, so antiviral therapy is available for influenza virus infection. It has to be started 48 hours on the onset, from the onset of the virus. Neuraminidase inhibitors such as zanamivir, all the ivir things, oseltamivir, peramivir. Okay, if you don't remember the drug name, just write neuraminidase inhibitors and imivir drugs. Okay, it is the drug of choice. Okay, for H1N1 strain, which is very common right now after 2009. And anyways, so the treatment was this, and the schedule for the treatment was that it would be given twice a day for 5 days. Now we'll come for the vaccines. Okay, now there are vaccines, as in there are 3 types of injectable vaccines for influenza virus. One is inactivated influenza vaccine, Fluzone. The example of that is Fluzone, inactivated influenza vaccine. Then something is cell culture based inactivated influenza vaccine, cell culture based inactivated, and recombinant influenza vaccine. These 3 vaccines are pretty important. You should remember the name: inactivated influenza vaccine and recombinant influenza vaccine. Schedule is basically single dosed administered intramuscularly to people except from the age group of 6 months to 8 years. In this age group you can't give this, and the 2 doses are required for this at least 4 weeks apart. Live attenuated influenza vaccine is also pretty good. The live attenuated influenza vaccine can be given to all healthy person between the age of 2 to 49 years of age, except in pregnancy. That's it. And thing is, the live attenuated vaccine is very much adapted to cold master strain, so it can grow in upper respiratory tract but not in the lower respiratory tract. Upper respiratory tract has a temperature of 33 degrees Celsius, but lower respiratory tract has 37 degrees Celsius, so it can't grow over there, and hence it cannot cause any severe complication or adverse effect in the lower respiratory tract, so it is pretty advisable to take this. The injectable ones are like part of the immunization program. So yeah, that's it. That was that injectable vaccine, and this was the live attenuated vaccine, LAIV. Now it's done. Influenza is done. Best of luck.